Harvey, get in your seat. Just kidding. Yeah, we won't put it. Okay, I'm going to bring people back together. Okay. We're going to start bringing people back together, please. Are there folks still out in the hall? Do you know? Pardon? Oh, okay. Amy, would you mind just letting people know we're starting? So welcome back. I hope that lunch was okay for everybody. And thanks again to all our docs who stayed around. I think it was really nice to have some informal time with them and just to connect with each other. So welcome back to the afternoon session. We have 2 sessions in the afternoon right now, and then we're going to break out into the small group peer-to-peer sessions. So hopefully everyone has had a chance to sign up for a group for that. So we're going to have 2 sessions now and then a break, and then we'll come back and we'll have the social workers break us into groups and take us to the different rooms. So the first session this afternoon is on recent research advances. And we are really lucky to have Scott Woodman here with us today. You've heard his name already this morning. He is really one of the up-and-coming stars in the research arena here at MD Anderson and really throughout the ocular melanoma community. And we're just so lucky to have you here today, Scott, to tell us about recent research advances. And Bita Esmali was going to join just briefly at the end of the session. We're not sure if she's going to be able to make it or not, so Scott may be Scott for the first part and then Bita for the second part, or Scott may be Scott for the first part and Bita will be Bita for the second part. But so stay tuned. So I'll hand it over to Scott.
Thank you very much. I was fortunate to participate in the patient symposium in Philadelphia, and it was just a wonderful experience. And so I'm really happy to be back and to talk with you all and introduce this topic. You've seen elements of things I'm going to talk about already from all the speakers. So hopefully this will be some consolidation. And then I was told never to give a talk after lunch. but I hope that these elements will come together, and we're going to go back to basic biology. So I need you to get those neurons firing. So targeted cancer therapy. You've heard a lot of this word, targeted. And what does that mean? Well, I think what it means is that traditionally you had a group of patients who had a histological type. They had a tumor that arose from a certain type of cell, a melanoma or a melanocyte in melanoma's case, and when a pathologist looked at it underneath the microscope, said, yep, it's melanoma, and so they were all grouped as such. And then they would go on a trial with an agent, and you'd have responders, hopefully, and non-responders. But that's where it ended, because there was no understanding of what the responders had that the non-responders didn't, why they responded versus didn't respond. And then even worse, in early phase trials, when you're using a drug, if there are no responders, sometimes that drug is stopped in its development because they don't think it has any efficacy, even though they were looking for just the maximal tolerated dose. So in the targeted therapy area, the idea is that even though you may all be of one histological group, there are subgroups that can be broken down molecularly, or green, red, and purple. And this has the advantage in that when you have trials, you may enrich to get more responders. And this was the case in breast cancer, where patients that had a particular receptor called HER2 and a drug was called Herceptin, that they enriched for that trial because those patients had that particular receptor and they had a positive trial. And then the other is, of course, is that you might enrich phase 1 trials to see if you get a signal that that particular subtype of the group will respond to that particular drug. So it's, this can be further and further refined based upon our understanding. Now, it's been you've heard already, it's a very exciting time in oncology, and the reason for that is represented on this slide. That is that we have targeted agents that are working, and they're working extremely well in certain cancers. Now, you all, unfortunately, melanoma itself gets lumped into a rare cancer type, and then uveal even further rare. But I'm here to tell you, rare is not bad. Rare is good because when you look at CML, for example, chronic myelogenous leukemia, and Gleevec was the drug that was shown to had efficacy at CML, there are only 4,800 patients a year in the United States that get CML. Yet there's been more money put into research of CML because there was the right drug and the right target, which was BCR-ABL translocation, that those two things together, if you can find the targets and you can find the drugs, a lot of energy and attention goes into that cancer, even though it is a very rare cancer. Same is true of gastrointestinal stromal tumor, or GIST, 4,000 to 5,000 patients a year. Same drug, they have c-KIT mutations. And I could go right down the list. Even lung cancer, which is one of the highest frequency cancers in the United States, only 6,000 patients a year have this particular genetic aberration, the ELK translocation. Yet this has become a very hot area of research because of that. So I think uveal, when you have a rare subtype like uveal, it means that it's typically a more homogeneous group. And therefore, unlike lung cancer, which probably has multiple subtypes that we don't even know about yet, or breast or colon, in uveal, I think we have fewer subtypes, and thus a purer molecular categorizations can be done. So cancer biology primer, this is the part I was saying, we have to go back to Bio 101. All right, here we go. So you have a cell, and that cell has a nucleus, and within that nucleus are chromosomes. And in the human cell, we have 23 pairs of chromosomes, and you get one from mom and one from dad. So chromosome pair 1, chromosome pair 2, and so on, up to the sex chromosomes, where if you have two X's, you are female, and if you're XY, you're male. So what is a chromosome? A chromosome is how the cell packages DNA. So here's a sequence of DNA, and I'll tell you what that is in a minute, and then it wraps around histones, which Sapta mentioned earlier. Histones are these proteins that they kind of swivel around, and it keeps on wrapping and wrapping and wrapping, and you're left with a chromosome, okay? So and you need that because the chromosomes all have to fit inside this nucleus. So that's what a chromosome is, bundled up strands of DNA. What is DNA? Well, DNA essentially consists of 4 molecules: adenine, cytosine, guanine, and thymine, and we just refer to them as A, C, T, G. And in the human chromosomes, those 23 pair I mentioned, there are 3.2 billion A, C's, T's, and G's. What is a gene? A particular sequence of A, C, G's, and T's, or a code. And the cell uses that code just like a code for anything, a code to get into a car, a code to, a telephone code. It uses that code to make a protein. And each human cell has about 23,000 genes. What is a protein? The protein is the workhorse of the cell. It's the machine. It's the thing doing the work. And so here's a kind of a representation of a chromosome. There's yarn here, and it's unfurled. And then here's gene number 1, and it's a particular sequence, A, G, T, C, C, T, G, A. That is a particular sequence that is not reproduced in the genome, and it codes for the protein insulin, for example. Here's another one, gene 2. Codes for BRAF, which is mutated in cutaneous melanoma, and so on. So each chromosome has a set of genes, and these genes being very particular sequences of A, C's, T's, and G's. All right? Well, the reason that we went through that is that because cancer is caused by changes in DNA, and you can have really big changes, as we have in uveal melanoma, where you can lose a whole chromosome 3. And that's called monosomy 3, okay? So there's only one chromosome 3 here. Or you can have small changes, as in this case, where here's the normal sequence of DNA, and then at this one base, where there should be a T, there's an A. And that one nucleotide difference, that one DNA difference, is sufficient to make this BRAF protein hyperactive all the time, always on, okay? So you can go from big changes in DNA to even the smallest little change in the DNA, and they can have major effects that essentially make for cancer. Everything that Sapna mentioned on that circular diagram of what a cancer cell has to do. So a chromosome, then, you can think of as like a phone book, where the genes are the phone numbers that have those distinct sequences of A, C, Gs, and Ts. And the hard part is, as a scientist, finding the mutations is like trying to find a typo in a phone book. You have to really dig deep and try to find it and understand the typos that matter, because some typos don't matter and some typos do matter. When I first started this was a laborious process, and I did this work like this, where you had these little ladders, and it was radioactive, and like this would be an A, so this would be like A, G, A, and you actually physically count them. Those are the Stone Ages. Now we have automated sequencing, where I literally give a tube to a core facility, and then they give me the data when they're done. And each of these little colors representing a particular A, C, T, or G. And this is called next generation sequencing, and it has revolutionized genomics because it allows for us to essentially have the ability to find every mutation in all the genes within a tumor. And that's a very powerful thing to have as a scientist and someone trying to create drugs to approach a particular cancer. And so by understanding the DNA changes that lead to a particular cancer, try to generate drugs that specifically target the mutation. So I remember this mutation, one little nucleotide change, makes this guy BRAF always on, always active, which it shouldn't be. But there are now drugs, small molecules, that can block that activity very specifically. And the proof positive of this is, here's a patient at baseline. Everything in blue is unfortunately cancer. And this is 2 weeks on vemurafenib, a drug that blocks BRAF. This is a patient with a BRAF mutation, and that's 2 weeks later. Just remarkable. And the black that you see there is not tumor. Those are actually the kidneys, because there's activity. Anywhere there's cells that are very active, the brain, the heart, the kidneys, and then this is the residual in the bladder of the dye, light up. So essentially, you're not seeing any tumor. Really remarkable. This is a patient with mucosal melanoma with a KIT mutation, and this is 4 weeks on dasatinib. Again, a small molecule that blocks that protein when it's hyperactive on all the time. And you can see the activity of the tumor markedly decreased in the pelvis of this patient. So these are the things that, as oncologists, we get very excited about, because it's just, to see it melt away in 2 weeks, unbelievable. And for patients too, I've even anecdotally, there have been patients that have gotten the BRAF inhibitor, and within a day or two, they tell you they feel so much better. Now maybe that's a psychological benefit, but I kind of have to believe it's real. So the frequency of mutations in melanoma was discussed earlier, and you were told that there are different locations where you can have melanoma, and based on those different locations, you have different gene mutations. So the typical skin melanoma that you're all familiar with has a high rate of BRAF mutations, NRAS mutations, and a low bit of KIT mutations. However, there's a form of melanoma called acrolentigenous melanoma that can be on the palms and soles, and those tend to have KIT mutations in a little higher, and mucosal melanoma also can have KIT mutations. Now I put the years up here to show you what year the mutation was discovered, so that you could have a sense of what was mentioned earlier by Tim, that in uveal melanoma we're kind of a little behind, but we're catching up quickly. Because in 2009, Boris Bastian's group showed that there were mutations in Jeana Keough in uveal melanoma. In 2011, Jeana 11 was discovered to have mutations. These mutations are mutually exclusive. If you have a Q, you don't have an 11. If you have an 11, you don't have a Q. And that's because they're redundant. They serve the same function, so it'd be redundant to have. And this is in 40 to 45% of patients have a Q, 40 to 45% have an 11, so up to 90% of patients will have either a Q or 11. In 2011, BAP1 gene was discovered to have mutations and in 45% of patients and in 85% of patients with monosomy 3. And then in 2013, just published is SF3B1, and this was in 18% of patients. This gene was mutated in almost exclusively class 1 patients. And so this may be a gene that's involved, it wasn't explicitly said in the paper, but it may be the gene involved in the class 1B subtype. And it was 16% in non-monosomy 3 frequency. And these two genes, mutations, tend to be essentially mutually exclusive. I think there was only one case where they overlapped, if I remember. So the MRF Cure-OM grant that was awarded to Levi Garraway, and we're actually collaborating with Levi on this particular project, to sequence every gene, like I was saying, that next generation sequence, every gene in greater than 65 uveal melanoma tumors to fully characterize the mutant gene spectrum. So this is what we know now. And then after this project is done, we'll have a very good sense of, are there any more genes that are mutated, and at what frequency are they mutated? And so we'll have a whole compendium, essentially, of the mutations within uveal melanoma. Well, are there other mutations? Well, short of what Levi and we are going to do, we and Ivana Kim at Harvard have both done similar analyses where we've looked at essentially genes that are mutated in other cancers in high frequency, and to ask the question, are they also mutated in uveal? And as you look down this gene panel here, from A, this is alphabetical, but you start at ABL1 and you keep going and you keep going all the way to WT1. 0 was the answer. That none of these very frequently mutated genes in other cancers were mutated in uveal. Again, arguing that uveal melanoma is a pretty homogeneous form of cancer where those mutations I mentioned to you earlier are the big players. We may find others with the next generation sequencing, but thus far it may be the case that it's a pretty parsimonious mutation landscape. So this is the proposed sequence of events. This comes from a review from Bill Harbour, where you have a melanocyte in the choroid or ciliary body. The early event is a mutation in Jeana Keough or GNA11, and this is what causes a nevus. And so when they've looked at nevi within the eye, they do have mutations in Keough. So a mutation in Keough or 11 is sufficient to grow a tumor. A little nevus. Well, I have a whole bunch of nevi on me, right? And you do too. And your nevi on the skin, believe it or not, have a high proportion of what? BRAF mutations. But you may never go on to have cutaneous melanoma. Likewise, most people who have nevi have a Jeana Keough or GNA11 mutation, but most people with nevi will never go on to have uveal melanoma. You need another genetic hit. And that other genetic hit that we understand so far is the BAP1 mutation for those folks who are classified as class 2. And this new gene that was discovered, SF3B1, seems to be associated with class 1. So maybe that's the second hit, potentially, there. There may be other genes involved, but this seems to be the progression so far. Now, these other chromosomal aberrations may also play a role, but a particular gene on those chromosomes has not been identified to date as being causative. And so you've seen a lot of these slides, a lot of these cartoons, and all I wanted to say about this is that Jeana Keough and GNA11, they signal through multiple pathways. So what happens in a normal cell is that you have a molecule that binds to this receptor in the membrane of the cell. This is the membrane of the cell. And then it tells the Jeana Keough to be activated. And then Jeana Keough activates all these other pathways to tell the nucleus to do something. Make this protein, stop making this protein. It's a regulatory signal. Now, when, excuse me, so Jeana Keough should not be on all the time. It should only be on when the right stimulus is causing the receptor to tell it to be on. In the mutant case, where the Jeana Keough is mutated, you don't need any of that apparatus. It's just on. And the reason is that mutation causes the Jeana Keough to look like it's in the conformation of always being on. So now all these signaling pathways are potentially on all the time, and that's a problem. So how, so how do we attack it? Well, invent a drug that blocks Jeana Keough. Invent a drug that blocks GNA11, just like you have a drug that can block BRAF. Well, there's a problem. The problem is that Jeana Keough and GNA11 structurally are different. In the inactive state, the molecule has a couple phosphates on it, and then when it's activated by that external signal that I mentioned earlier, it's activated by gaining a third phosphate group. Then it has an intrinsic, meaning an enzyme within the molecule that cleaves that phosphate off and returns it to the inactive state. So you have to have that intrinsic GTPase, that enzyme within it, to inactivate the molecule. Well, the mutation in Jeana Keough and GNA11 make that enzyme not able to function. It cannot cleave off that third phosphate. Thus, the molecule is on all the time. Then you might say, well, invent a drug that just cleaves off the third phosphate. Well, the problem there is that this is a very common, ubiquitous looking target. So it would be very difficult to have specificity by doing that. So there have been other possibilities. One possibility is, well, all of these other downstream molecules we have drugs for. Everything I've put in a red box, there are drugs for these. And that's what's happening now, and that's what Sapna showed you earlier, is that this is the low-hanging fruit. We have drugs for MEK, we have drugs for RAF, we have drugs for PKC, and therefore, that's what the trials have been designed to use. Right off the shelf, let's do it. Another possibility, and this highlights the other grant that was funded by Chromosome to John Sondek, is that he studied Jeana Keough and GNA11. Oops, there we go. He studied Jeana Keough and GNA11, and he found out that when it binds to its binding partners, its immediate binding partners, it takes on a very similar structure every time. And so he's trying to discover molecules that just disrupt that binding. Pretty smart. And that's why I got the grant. So that's and he's been had a wonderful science paper a couple years ago and is developing that as a strategy. Wonderful. So that is not a drug yet. Can't pull it off the shelf and give it to somebody, but it's a very good place to start to try to invent a drug. Then you may have heard of c-Met and IGF-1R. These are receptors that are thought to be highly expressed in uveal melanoma. And the growth factors, hepatocyte growth factor and insulin-like growth factor 1, are in the liver. They're in other places too, but there are in the liver. So there's been a thought that maybe these are enhancing uveal melanoma's ability to survive in the liver and proliferate. And so studies have been looking at these too. So that's the challenge with Jeana Keough. We can't directly target it, but we can target its downstream mediators right now, and we're working on targeting its binding to its partners. Now what about BAP1? Well, BAP1's a challenge. Why is BAP1 a challenge? Because when it's a loss of something, it's not a gain of a function, that you're overactivated, it's a loss of a function. So here you can see staining of tissue from a class 2, class 1 patients, and they have BAP1. But class 2 patients, it's not there. So you say, well, that's easy, just put it back in. Well, the issue to date has been it's been very difficult to do gene therapy, to get a gene into every cell that needs it, have it express the right amount of protein, reverse, right? So that's going to be very difficult to do. But you can try the same strategy as you try in Jeana Keough. That is, there are downstream ramifications from BAP1 being lost. And we know what BAP1 does to a certain degree. We're still learning. But one thing it certainly does is that it serves as a transcription factor regulator. All it is fancy talk for it turns on genes. It gets down and is a stimulator for turning on genes. So if you lose it, those genes aren't turned on. And if those genes aren't turned on, maybe those genes are suppressive to tumors. Chromatin remodification. So this is those histones I told you that spin the DNA and wrap it. Well, those can be modified by all kinds of tags. Let's see if I did this. No, I didn't do it right. Anyway, they can be modified by all kinds of tags. One of those tags is called a ubiquitin, and BAP1 just lops that off. Takes the ubiquitin, takes it off, takes it off. Well, if you lose BAP1, that histone is going to be ubiquitinated, and that's been shown to regulate genes again. This is all about regulating whether genes are turned on or turned off. So BAP1's a tough issue, but once we understand better what signals are downstream of the loss of BAP1, maybe we can block those. Well, one way you can research that is in a mouse model. So what happens in a mouse when BAP1 is removed, or we call it knocked out? This was just published in Science. The loss of BAP1 causes myeloid transformation. So these mice get a form of what's called MDS, myelodysplastic syndrome, a particular kind, chronic myelomonocytic leukemia. First of all, when you knock out the gene, they die in the embryonic stage. So BAP1 is important for development. Okay. So when that happens, what you can do is you can make a mouse that you turn off the gene when it's an adult, and that's called an inducible knockout, right there. And this is what we call the phenotype, or what it looked like when it happened. So why didn't they get uveal melanoma? Why didn't they get mesothelioma, which is associated with BAP1 mutations? Why didn't they get renal cell carcinoma, which is associated with BAP1 mutations? Because a mutation in isolation, a gene that's mutated in isolation, says nothing. It's all about the context in which that mutation happens. So in uveal melanoma, you probably need a Jeana Keough or a Jeana11 mutation in addition to the BAP1 loss. In this mouse, they didn't have Jeana Keough or Jeana11 mutations. So this is purely what happens in a BAP1 loss situation. But it does tell us something. MDS is thought to be a epigenetic disease, meaning that the changes that drive MDS aren't necessarily from mutations. Well, they are from mutations, but in addition to mutations, it's from things that happen to the genes that have nothing to do with switching the A's, T's, C's, and G's. And I'll show you what that means. So here again, in yellow, or let's look at this. So in red is the DNA wrapping around the yellow, which are histones, these proteins. And these histones have these little parts coming off, and you can do all of these different modifications. Not important to know what they are, but there is the ubiquitin one I mentioned. Another thing that you can do that's epigenetic, you're not changing the DNA, you're not changing an A to a T, a C to a G, whatever. You're just lumping on a methyl group, it's a particular type of molecule, and then you change the ability of that gene to be made. So these are all modifications, epi, above, right, genetic. These are all modifications that can happen to a gene without actually changing the code. Okay, so is that, so that's what the mouse may be telling us, that BAP1 is really important for epigenetic modifications, and we kind of know that. And so that would change the drugs that we would use, because we don't use, we have drugs that can be used for diseases that have particular epigenetic modifications. But it's complicated. So the overall challenge is that it is really I love this diagram because it's a network. It's a network. And there's Jeana Keough right there. So let's say Jeana Keough's mutated and it's causing all of these downstream effects. Okay. Well, what happens when I block one of those downstream signals, like MEK? There's MEK. I block MEK. Well, we run into the whack-a-mole problem, right? I block MEK, but then the circuit goes in a different way, right? And so this goes up, this goes up, this goes up. And so we have to understand what happens when we block those downstream molecules because we want to understand why the cells aren't, they may slow them down, you may get a smaller tumor, it may stay stable for a long time, but eventually there's a recurrence, right? And so or a progression. So we have to understand all of that signaling network that's going on. And the good news is we can meet that challenge. I already told you about genome sequencing. We can know all the changes that happen in every gene. We can know how those are expressed in RNA, and we can look at the proteins. And all you're looking at here with all these colors is red is up and green is down, and every one of these vertical lines here is a particular RNA, and every one of these down here is a patient. These are all different proteins. These are patients or timer. And the point is that we can account for a lot of the network diagram. And we can understand, well, what happens, let's look at the tissue before a person goes on a drug. Let's look at the tissue while they're on the drug. Let's look at the tissue if they progress. And let's understand all of the different ups and downs that are occurring that are allowing the cell to continue to grow. And so I mentioned the next generation sequencing. There is a project called the Cancer Genome Atlas, TCGA. Get it? TCGA, right? So those are the letters of the DNA. And this is a very busy diagram just to say that, and Sarah's going to talk about this in the future, that there's an interest in doing a project in uveal melanoma. And that project would be looking at the gene mutations, another thing that can happen, an epigenetic thing, DNA methylation, mRNA expression, microRNA, I haven't even talked about, and protein expression as well as clinical data. And then integrating all that information so that we get the complete and global view of uveal melanoma. And this is being done in other cancers now, and the thought is we can do this in uveal melanoma as well. I'm going to skip that. Dr. Keough talked to you about immunotherapy, so I won't talk to you more about immunotherapy other than to say that the immune system has gas pedals and brakes. And as you look down the T cell here, you look down at the names of these, and they're either plus or minus, meaning plus meaning growth and minus meaning inhibitory, so brakes versus gas pedals. And ipilimumab, or Yervoy, blocks this guy here, which is a brake. So if you block a brake, you make it more active. So it makes T cells more active. PD-1 is also a brake, and there are drugs in development to block PD-1, which would, again, activate the T cells. So it's a gentle balance of taking off brakes and putting on gas pedals. And so this, I hope, kind of represents for you that there are a lot of potential options in gas pedals and brakes in immunology, and we're just beginning to modulate them. And the ultimate sort of vision is to combine these things. All those targeted therapy options that I was showing you earlier, all those pathways, understanding them, understanding what's up, what's down, what we can block, in addition to immunotherapy, teaching T cells to be more active and eat tumors like Dr. Keough showed you earlier in that movie, and potentially combining these in rational ways that make sense, that are well tolerated and kill tumor. That's our ultimate vision, and it's becoming a reality. We are already doing it in cutaneous melanoma, and as Dr. Keough said, there's no reason to think from an immunological perspective that uveal melanoma should be all that different given the markers that it presents. So our conclusions are these, that the current state of knowledge is that we have a small set of high-frequency mutations, and they appear to be the major mutations, but the challenges are that we don't have anything that can directly inhibit them, and with BAP1 we can't replace it as such. So we have to use our knowledge of the mechanisms which are at present incomplete. But as we learn more about how these genes work when they're on and off, we will be able to better approach them. And single agent treatment may work for some folks, but ultimately I think a lot of us look at this almost like HIV. You know, it took a 3-drug combination to finally short circuit the virus. And in this way, in cancer, it might take the right combination, the right nodes within that network diagram that I showed you to hit the right targets to short circuit a cancer cell. And those are the opportunities, especially in uveal, because we have excellent markers to predict who will get metastatic disease. So this is when I show people, the class 1, class 2, monosomy 3, disomy 3 prognostic test, my colleagues, they're blown away. We don't have that in many other cancers. And what that allows for is you to select patients who are high risk for metastatic disease up front and get them on a clinical trial, an adjuvant trial. Again, very homogeneous group, which is good for clinical trials because you're treating the same entity, unlike lung cancer where you might be treating 10, 15, who knows how many subtypes of lung cancer. More oncology drugs in the pipeline than ever. You saw that today with Dr. Hong, the number of drugs coming along. Combinatorial therapies may show greater efficacy, and immunological therapies alone or in combination may be efficacious. And this is where we're at right now, is to do that in a rational way. And that is the end of my talk. And I have to unfortunately leave by 2 because my 7-year-old is giving a dance performance. And but I'm going to come back tomorrow and I can answer questions now, but then I'll be back tomorrow as well. Bita, do you want to?
I'm flexible, but
okay.
I can go next, or you can answer the question if you need to go.
I mean, this session ends at 2 anyway.
Oh.
Whatever works.
Okay, I can answer questions and then, yeah.
Okay,
Okay.
so why don't we'll take 5 or 10 minutes for questions for Dr. Woodman, and then we have Dr. Esmali here as well. So if you have questions, if you want to go to the microphone, can you go to one of the microphones, please?
I know that was a lot of information.
I know that Dr. Patel was this year working on the ipilimumab. Are you still involved in that aspect, or have you moved on from it?
Yes, so that's an
So
active...
you're still with her and doing all that stuff?
Oh yeah, we're a big team at MD Anderson, and Dr. Esmali and Dr. Gambos, the ophthalmology group, and then myself in the research, and we have other researchers, Liz Grimm and Chandrani Chattopadhyay, and so we have a big unit, Now, just to drill down a little bit on your question, so for that trial, we are trying to do as much of the, what we call correlative studies, as much of the blood work as we can, tissue biopsies as we can, because at the end of the day, we will never understand this disease or any other cancer unless we can take, multiple snapshots in time and drill down. Yes, ma'am.
It's my understanding that every delegation of cancers around the body, all the metastases, set up their own little nest and have their own different mutations. So how could any one system knock them all out?
It's a great question. So one of the things I did not talk about, but is a very, very important issue in cancer therapy, is what we call the microenvironment. So a uveal melanoma cell that grows in the lung versus the liver versus a bone, that's a very different environment with a very different selective pressure and therefore potentially different mutations. And we're actually studying this right now in all cancers at MD Anderson to ask the question, how different are the different tumors within different sites of the body, or even within the same site if you have 2 different tumors in the lung. And so far, it looks like there's a lot of commonality, which is good news. That is that you don't lose particular mutations. So they'll all have Jeana Keough, they'll all have GNA11. There are examples where that's not the case in some other cancers, but thus far in uveal it's been true. And so we're hoping that there's enough commonality from that original clone that's shared among them that we can short circuit it. So the good example of that is that CT scan I showed you earlier of the BRAF mutant tumor responding to a BRAF inhibitor. You didn't see, you just saw the whole thing going because they all had the BRAF mutation. They may have other mutations, but that was the important one. Right. Thank you very much.
Okay. Okay, I'm going to introduce Dr. Esmali while we're loading her slides. Dr. Esmali is professor of ophthalmology here at MD Anderson and is going to speak to us a little bit about the importance of tissue banking and what can be done with tissue banking and how that can help some of the research that you've just heard about from Dr. Woodman. And then we'll have time for probably some questions at the end of this session too before we have our patient caregiver panel. Okay, thank you.
It's my pleasure. Thank you very much, Sarah. This is a great turnout. I'm really happy to be able to talk to you a little bit about our tissue banking and blood banking efforts and how you can potentially consider participating in such protocols. Let me find the right talks. Just a second. Let's see. This one. So, as an ophthalmologist, of course, we probably are the entry point for many of the patients with the new diagnosis of uveal melanoma. And I think this morning, I'm sure you heard a lot about various different trials that have already taken place or are in progress. And one of the ways that patients can contribute to advancement of our understanding of this disease is indeed by participating in clinical trials. A couple of really basic facts that I know that you all, this morning in the Uveal Melanoma 101, you probably understood this or this was highlighted. But one major difference between uveal melanoma and other forms of melanoma is that it really is a clinical diagnosis, meaning that we don't really perform a biopsy to make the diagnosis of uveal melanoma. It's based on our clinical examination, based on fundus examination, perhaps ultrasound findings that we make this diagnosis. Therefore, by definition, tissue is scarce because we don't really do a biopsy to make the diagnosis of uveal melanoma. So relatively scarce tissue in terms of primary eye melanoma tissue. So, and then there have been major clinical trials in the last several decades that have demonstrated that various different types of local treatment don't really make a difference in terms of long-term survival. Therefore, most patients elect to have eye-sparing treatments. So enucleation, perhaps when it used to be the primary mode of treatment of uveal melanoma, it's less and less so to the point where it's estimated that perhaps less than 50% of the patients elect to have an enucleation. Enucleation, of course, is removal of the eye. The point is that there is a relative lack of tissue for research and for gaining new insights. And of course, we do need better outcomes. As with any other cancer, there is a need for better survival outcomes. We only have metastatic disease that is clinically detectable in less than 2% of patients that present with a new diagnosis of uveal melanoma. Yet, approximately 30 to 50% of patients eventually develop metastatic disease, and at that point, survival is really quite compromised. So there is a need for better outcomes, therefore a need for research. And as you probably heard during much of today, a lot of the understanding, a lot of the molecular understanding and insights that we have gained is based on access to tissue and blood. So that with the ultimate goal that we can maybe someday have targeted therapies that are based on molecular characteristics of the particular tumor that a given patient has. So this sort of individualized treatment that we hope to be able to offer someday very much hinges on access to tissue and perhaps blood. And of course, ultimate goal is to improve survival. Now, a few words about just unique things about uveal melanoma that make participation of patients in such protocols very important. For example, one aspect is that when an eye is enucleated, because the eye is filled with a gel-like protein called vitreous. It is typically, it's not possible to process that tissue immediately and get a frozen, snap frozen piece. And for much of what you heard today, we need access to fresh frozen material to do the appropriate molecular study. So just by definition, when we want fresh frozen tissue, there has to be a protocol that moves away from standard of care. So we have such a protocol at MD Anderson. There are several centers throughout the United States that do the same. This means that when the eye is enucleated, we get permission from our patients ahead of time to be able to take a small portion of their tumor, freeze it immediately for future molecular studies, and we also get their permission to collect their blood, again, for future molecular studies. And I hope to end my talk by showing you how that has really paid off. Today, we've had a protocol in place for the last 12 years, and because of this type of prospective reaching out to patients and getting their permission to use their tissue and their blood, in our protocol, we are now able to participate in very large-scale and meaningful, in-depth molecular studies that hopefully will teach us how to do targeted treatment for this disease and ultimately improve survival. So the point is that the getting fresh frozen tissue is a departure from standard of care. So it is something that the center that you go to for treatment for uveal for eye melanoma, for the treatment of the primary uveal melanoma, has to have such a protocol in place because it is a departure from standard of care. And we don't need to get into too much of the technical aspects of this, but there are specific steps that have to be taken in order to be able to get a fresh frozen sample out of an enucleated specimen. Dr. Michelle Williams is the pathologist at MD Anderson that does this for us. She has developed a specific protocol that she follows to make sure that we don't compromise the standard histologic evaluation of the specimen, yet get enough tissue to do future research and bank it for future research. Yeah, I think I already mentioned this, that we try not to use up the tumor. We try to save enough for standard histologic evaluation and then freeze the rest for future studies. And the other part, the other important point about unique to uveal melanoma is that unlike cutaneous melanoma, most of the patients have liver as their primary and most common site of metastasis. Therefore, access to liver tissue is also key because it is commonly believed that perhaps there are molecular changes, genetic changes that take place from the time that the tumor is just limited to the eye to the time that it becomes widely metastatic. So again, we have a limitation because liver is a relatively difficult to get to organ, and it's a critical organ, so doing open biopsies, large biopsies is difficult to do. So we are limited to fine needle aspirations or core biopsies. Again, participation in clinical trials that allow us to use that tissue for future studies will really go a long way in improving our understanding of what leads to metastasis in uveal melanoma. So again, scarcity of tissue and importance of participation in clinical tissue banking protocols. So I mentioned already fresh frozen samples is important. It's a departure from standard of care, and we need to strive to have that available for all patients who undergo enucleation. Blood is another very precious resource. Prospective collection of blood at various different time points, including at initial diagnosis and then throughout the course of follow-up, is very important. And access to uveal melanoma cell lines is really a very precious resource. Cell lines, Dr. Woodman mentioned, and I'm sure other investigators throughout the day have mentioned this, cell lines are essentially tumor cells that we plate out and allow us to study in the laboratory, perhaps response to various different drugs. When the new drugs come along, the uveal melanoma cell lines allow us to study these drugs in the laboratory. So again, establishing uveal melanoma primary and metastatic cell lines is a real precious resource that we can potentially develop when we have access to enucleation specimens. So why should a patient participate in blood and tissue banking protocols? Again, to summarize what I've already mentioned, there's minimal to no additional risk to an individual patient for participating in his trials. We really don't compromise the standard of care. These are patients that already have elected to have an enucleation, which is removal of the eye, due to various reasons, including the size of the tumor, patient preferences. And just really cannot emphasize how precious this resource is for our understanding of the genetics of this disease, and for our ability to develop new targeted treatments that hopefully will lead to better outcomes, ultimately better survival, both in the adjuvant setting and in the metastatic setting. This is the era of adjuvant trials for uveal melanoma. While we don't really have great drugs yet that can easily target BAP1 or other loss of BAP1 or GNA11, Jeana Keough that Dr. Woodman mentioned, I think the future is bright in that the more we understand about molecular characteristics of uveal melanoma, the more we can more rationally design trials that will offer treatments early on in the adjuvant setting. And also, while not yet, we are not yet there yet, I think someday, hopefully, it will be serologic and blood tests that will lead to earlier diagnosis of metastatic uveal melanoma, and or better diagnosis of who has the high-risk tumor. And these are, while we may not be there yet, collecting blood now, hopefully someday will allow us to do that. So I think these are, again, I'm really just probably repeating myself, but I can't emphasize how much a patient can contribute by just by enrolling in tissue and blood banking protocols. So I want to finish my talk by highlighting this collaborative multi-institutional effort that QRM was very critical in organizing and arranging by putting us all in touch with NCI. So the Cancer Genome Atlas project for uveal melanoma is a new project that is sponsored by NCI. It is just getting off the ground. TCGA, or the Cancer Genome Atlas, is a major investment by NIH after a pilot project in 2009. Several $1 million are now dedicated to this project, and uveal melanoma is one of the newest cancers that is included in this project. And this is a comprehensive genomic sequencing and characterization. And for this to take place, we need fresh, frozen tissue and matched blood. And even though this is happening in 2013, the only reason we are able to participate in this project is because we have been collecting frozen samples and blood for the last 12 years. So again, the value of participation in banking protocols. So this project is probably going to be getting off the ground this calendar year. The goal is to have 100 matched blood and frozen tissue samples by the end of 2013. And there are several centers throughout the United States that have expressed an interest. Our MD Anderson is participating. There's the Liverpool Ocular Oncology Unit in the UK, Wills Eye Hospital, Cleveland Clinic, University of Oregon, and many more that are coming on board with this trial. So I just wanted to end by giving you my contact information. If you want to know other centers that have prospective blood and tissue banking protocols, I'm happy to put you in touch with the right folks in where you live. And of course, if you have any other questions about any of this, I'm happy to entertain them now.
Yes, I have a sample at UT Southwestern. It's about 3 months old. I hope it was preserved properly. Would it be too late to enroll?
No, I don't think so. First of all, for the blood component, for blood collection component of at least our trial here at MD Anderson allows for participation even after you get treatment for your primary uveal melanoma, so you can still participate in that component of the protocol. And as far as tissue preservation, paraffin embedded tissue, which is basically the the formalin-fixed enucleation material. That's still useful in terms of getting information from the fresh frozen, snap frozen or OCT frozen samples may or may not be available on your tumor. I can't, that depends on exactly how they process the tissue.
Okay, I know that a portion of the sample was prepared with the wax, with the paraffin, and sent to Castle Biosciences.
Sure.
Should maybe I contact them? Or?
No, well, so most of the times, unless you signed a consent form for participation into a protocol or anything like that.
I was never offered.
Yeah, so then your tissue is most likely archived in the center where you had your enucleation in their pathology lab.
Right.
Those blocks are still available there. We can talk a little bit offline. If you want to participate, like in our trials, you would have to become a patient at MD Anderson, and you can still participate for the blood and serologic collection component. The fresh frozen sample, that may be difficult to do retrospectively.
Okay, well, I mean, becoming a patient at MD Anderson wouldn't be an issue for me. I'm just a little frustrated that something like this that's been around for 12 years wasn't offered to me initially when the eye was removed. It seems kind of odd. You think that the pathology lab, all the different pathology labs would be on board.
No, that's a good point. I also didn't want to mislead. I mean, this is not, of course, a treatment trial. This is a tissue banking protocol. So you weren't, in other words, you didn't miss out on a treatment. But yes, it would have been a good resource for us. But it is difficult to logistically get tissue from another center and freeze it here, so that probably is the limitation. That's why it wasn't really offered to you as a readily available thing, I would imagine. Do you have other comments about it?
I was just, one comment I was going to have, I don't know, we're almost out of time, so if you want to mention something about this, or I was going to talk a little bit more about it tomorrow, but one of the things that we're doing that I was going to give in our scientific update tomorrow, I'm going to talk a little bit more about the TCGA that Dr. Esmali mentioned, but as part of the Chromosome initiative, we are developing, we're trying to develop a virtual tissue bank, which would sort of speak to this problem where different centers have tissue at their institutions, but that we'll be developing sort of a virtual way that all the centers will be able to communicate. And so it's not there yet, but we're working on it.
It's a very good goal.
And Dr. Esmali has been really instrumental in helping us try and reach that goal. So I just want to mention that.
No, good point. Yes.
All right.
Thank you.
Thank you very much, Dr. Esmali. Really appreciate it. All right. Oh, yeah.
The little green thing on top.
Okay. Okay, just a few technical things. So maybe the folks on our next panel, if you want to start making your way up, we'll just kind of have a few technical things to do and then introduce the next panel. Can we get our other slide back up? Here, right here. Just gotta click on it. No. Okay. And Sarah. Okay. So I'll just give a quick introduction and turn it over to you, and then we'll introduce them. Yeah. Okay. Okay. All right. Thank you. So actually, yeah. Do you want to sit here for a minute, and then I'll have you come up, and then you can go back and forth. Okay. 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 So while we're getting people up here, we've had a long day so far and a lot of medical information thrown at us. So I just thought in the mindfulness sense, maybe we should all just, I don't know if people feel like getting up and stretching for a minute or just kind of taking a deep breath, but that is sort of, that's the end of the scientific presentations for today. So a lot of really good information. I feel a little overwhelmed, so I'm not sure how you guys are all feeling. But we're going to sort of switch gears here a little bit. I am really honored to present the next panel. I mean, I've been honored to present the physicians all throughout the day, but I have to say that some of the people who inspire me the most are all of you. And this is the first of a couple times that we'll be hearing from patients and caregivers. And so it's just a true honor for me to welcome you all up here as sort of representatives from our community, but these are a few stories. We know we all have our own story. So I think it's important to tell our stories and to listen to others' stories and to share. And that's what this panel is about, and that's what the afternoon will be about as well. So on today's panel, we have Butch Ewing, who some of you know, sitting at the end here. Esther Damaser in the middle, who's also on the Chromosome steering committee. And we have Jeff Mark. I will let them further introduce themselves when they come up here. But we are also very lucky to have Jeana Keough, who is a social worker here at MD Anderson. And she's going to be involved with our small groups, the peer-to-peer groups directly after this panel, but will be helping to facilitate this panel as well. And so I will turn it over to Jeana, and I'm just really grateful to all of you. So thank you.
Good afternoon. So I just want to second that it definitely is a privilege to have the opportunity to learn from your peers, from people who are in the same place, caregivers and patients alike. And so our hope is that you'll have moments where you can identify and say, yeah, I've experienced that, or yeah, I've felt like that too. But we also want to validate that there may be times where as they're speaking, you may have had a different reaction or a different experience, and that's okay too. And so either way, we want to join with them in their journey, hear from them, take the opportunity to learn from them, learn some of the wisdom that they've gotten along their journey. And then this will give us a perfect segue into our small groups later. So if you have different reactions or questions or thoughts, we want to give you an opportunity to discuss that further in a group of your peers too. And so the structure will basically be, we're going to give each of them individual time to talk about their different story, and then we'll have a question and answer session at the end. And then after that, we'll divide out into our small groups. So on the list that you signed up for this morning, we'll have different social work counselors available that'll have the name of the list, and then we'll take you into the different rooms so that you can have that specific time. So we'll go ahead and get started on the panel because they're really the treat this afternoon. Did you want to start first? These are on it.
Oh.
I mean, either way.
Well, hello, my name is Butch Ewing. I'm sorry? Oh. Hello? Thank you. What's interesting, I love irony, and what's ironic is I know that I look like a pirate, and I get that a lot. People say, you look like a pirate, and most of those times I don't really want to explain it to those people, but you guys know why I look like a pirate, and I want to explain it to you, so it's kind of funny. I was diagnosed in 2008. I had a 14 millimeter in diameter by 6 millimeter in height, and I opted for the plaque therapy, and I got that a month later. And since then, my vision has deteriorated to the point where I can barely see, but just enough for it to kind of have a strain, and it gives me headaches, so I wear the patch. Plus, it makes me look tough, And being a man of 5'5", you look for ways to look tough. But yeah, I initially noticed something. One day I walked outside to go to lunch, and I thought I had an eyelash in my eye. And I was, trying to get it out, and didn't think too much of it, except it never went away. I noticed that I couldn't feel it, but I could see it. I stood in the shower trying to get it rinsed out. I remember asking, my family and friends, hey, can you see anything? Because I can't get this out. And after several days, I decided, it's been about 10 years or so since I've been to the eye doctor. Maybe I should, go check it out. So I went to the eye doctor at lunch one day, and they did some tests, dilated my eyes, and looked in. At that point, the ophthalmologist said, would it be okay if maybe I had one of the other doctors come and look? And I'm like, sure. And then after she left the room, I thought, there's only one reason why you want another doctor to come and look. It's the freak show, you know? And so I think, okay, well, how is this going to go? So he comes in and looks, and within a second, I mean, he didn't even really have time to look, in my estimation. And he says, well, we're not exactly sure what this is, but we think you should go see a specialist. And I said, okay. And he said, well, you might as well go ahead and go now since your eyes are dilated. And all I heard was, go see a specialist now, you know? So I'm like, okay. So I jumped on my moped and with my eyes dilated, drove downtown. I had no idea, right? Yeah. So I get to the retina specialist, and this place was the freak show, in my estimation. People there with jackhammers in their eyes and eyes hanging out. I mean, it was just, you know, the worst of the worst. And I'm sitting here thinking, what am I doing here? You know? I finally get in. They start doing some tests. They tell me they want to do a sonogram, and I think, what do you think, I'm pregnant? You know, I lift up my shirt. I'm like, go for it. And they're like, no, on your eyes. Like, you think my eye's pregnant? How does this work? So, you know, this is between us, though. Don't tell any of my homeboys that I had a sonogram, okay? I'll lose my man card, all right? So they do the sonogram. They do tons of pictures, and then the retina specialist comes in. He sits down, and he says, well, what you have is a mole in the back of your eye that's turned to melanoma. Hopefully it hasn't spread to your liver or lungs. Of course, we'll check that. Now, back in the old days when I used to do this, the only forms of treatment was radiation or enucleation. And your tumor, I tell you, is so big, you're going to have your eye removed. So pretty bad news, huh? Yeah. And I said, well, at least you didn't say I'm about to die. And he's like, yeah. I don't remember anything until I was in the parking lot. I'm standing in the parking lot, and whenever I finally be the producer and star of my life film, this shot will be one of the shots that I want to make sure to get right. Because I saw myself outside of myself. And it was just as the camera angle went up and over me. I'm just standing in the parking lot, not knowing what to do. And then the busyness of Houston is all around me. Because I'm from Houston, by the way. So at that point, I call my best friend, and I said, hey, you know, here's the deal, and remember that eyelash that I couldn't get out, and here's what the deal is. And I said, do you think that should I go home and tell my wife, or should I just call her right now? I mean, I only went to the eye doctor at lunch, you know. And he said, yeah, you should definitely go home and tell her, right? So we go home, and we break the news to the family, and then at that point, we decided to start getting some other opinions, starting to see what else the other options were out there. We found a great doctor that ended up going with. And what's interesting is I told him, this guy, first of all, he is a few days shy of 100 years old, literally. I mean, this guy is old, which I wanted. I wanted somebody that had some gray hair, some experience, some time under his belt, you know what I mean? And I told him about what the retina specialist had mentioned to me. And his response, what I thought was quite wise, he said, you know, no one is prepared to hear that you have cancer, and no one's prepared to tell you. And I thought that was pretty wise. It still was terrible bedside manner on the retina specialist, I feel, but still it helped me to understand in some way maybe what that must be like as a physician. After that, you know, I started, you know, I went home immediately and told the family. And the first thing that I did, I'm a programmer, a web programmer for a living. That's what I do for money. So I went online, started looking at the information online. And as all of you know, especially 4.5 years ago, that information was pretty grim. There wasn't much hope out there. I couldn't find a single person in what I could find. I couldn't find a single person alive with this disease. And that was very tough. And so from then I decided, well, you know, that's what I do. I put things on the internet. So I decided to start putting things on the internet that maybe was a little more hopeful. Maybe finding more information and distributing that information. And that's what I've done. And I on Facebook, I've I came across an eye cancer group that was there. There was maybe 3 members. Didn't seem to have anything really happening with it. The original moderator didn't tend to it long enough that Facebook even said this group is now open if you would like to moderate this. So I did. And then that group has continued to grow, right? And it's just another resource. And to me, I think that as many places as possible that we can have information about ocular melanoma on the internet, the better. Because we can start to really populate and flood the internet with that information. I think raising awareness for this disease is one of our biggest obstacles and greatest goals. But I'm hopeful. I'm optimistic and I'm hopeful, especially after hearing a lot of things today. The future is bright. And one of the things that I've learned from this process, which is I think what I'm supposed to say, of what I've learned from this, is I've learned a lot about myself. I learned a lot about my faith. I've learned a lot about what it's like behind the curtain of Oz at a hospital. And I was just mentioning last night, as a programmer, I get to see how things are made. That a lot of people wonder, how do they do that, right? And I see that nothing is there by accident. And the more I learn about science and the more I learn about God, the more I realize that none of us are here by accident. And I don't understand it, but I know that there's some purpose. I know that I was in Dallas, living in Dallas, and by my estimation, the will of God moved me to Houston. And I changed jobs. I sold my business to another company to only 6 months later be diagnosed with cancer. To me, it was divine providence that I was able to have insurance to pay for all this. And so I'm thankful for that. So that's my story.
Catastrophe. If I come into a room, expect catastrophe.
Okay, so there's this light here. We have to use it over here if you want.
This is the
Yeah, this one's on.
microphone? Okay. Hello, everybody. To both my new and old friends, I'll ask you to forgive me. I'm going to read my speech because I don't have enough brain cells to remember it all. And I've also had a very long career with this cancer. It'll be 33 years. And in a way, the worst part is that I haven't done anything to merit it. I'll tell you right up front, I have no secret bullets. I've had a lot of treatments, and we'll talk about that later. But none of them is a secret bullet for anybody, and all of them are things that you guys have had before. Also want to tell you I have miserable eyesight, so this may not be a pleasant experience for us. Okay. I've been living with cancer for an awfully long time, as I just told you, and so I've had to make room for him, cancer, in my life. It has become almost a relationship, cancer and me. How can I describe this relationship? Dysfunctional? But what an understatement. We are, after all, trying to kill each other. But because I know that cancer is part of me, I say, to paraphrase Pogo, I have met the enemy and he is me. Though cancer is my evil twin, he is in the family. So I tell him, like the Jewish mother that I am, how can you do this to me? Very effective, except not with this evil twin. My cancer has responded by being easier on me than it has been with most people. But our disagreement is hardly over, getting more fierce as the days go by. I certainly have not given up fighting, and I know he hasn't either. So we are getting into a brawl here. 33 years ago, this monster arrived in my right eye. At the time and for a long while afterwards, I had no idea how monstrous he was. Cancer of the choroid layer of my eye. I didn't even know I had a choroid. Take my eye out? What are you talking about? Here was the first assault my cancer monster attempted to make upon me. I could hardly believe the fighting spirit it provoked in me, because my nature is to be compliant, obedient, good girl. I do what the doctor says. But my nature is also to learn about reality, evaluate it, assess the situation, and then act. I managed to avoid enucleation because the tumor was small enough and because I could find no evidence that enucleation would solve anything. So I did not have my eye taken out as the doctor I originally saw demanded that I had to. This was back in 1980 when it was the way you treated ocular melanoma. Instead, I found another doctor and received photocoagulation, which is a kind of laser. Maybe the tactic of Jewish mother guilt worked because my evil twin quieted down and hid for 8 years, after which it began to grow again, but only in the eye. This time I knew a bit more about researching the options and had brachytherapy done, which many of you have also. My doppelganger, my evil twin, was again in submission. Our relationship was in equilibrium. It took most of my eyesight, but I took a new lease on an appreciation of life. Time passed. 22 years passed, during which our relationship faded away. At least for me, I didn't pay any attention to it. I continued an ordinary life, seeing children grow up, go to college. My husband and I retired. I pretty much felt as though I had cowed or persuaded this evil twin to go to sleep or leave this relationship altogether, which would have been nice. I only thought of him when I was having follow-up checks. No doctor warned me to look for spin-offs, for metastases of this disease. But I knew that cancer could spread to, any cancer could spread to other parts of the body, and even though I was having regular eye checkups, I thought somebody should be looking at the rest of my body to see what may appear there. Following my own common sense, not any doctor's recommendations, I went to a medical oncologist to ask why I was not receiving follow-up surveillance. After turning white and asking incredulously, "You mean nobody is following you?" he ordered annual CAT scans, liver function tests, chest x-rays, and mammograms. How poorly equipped I was to do battle with my evil twin. I didn't know that once it had metastasized to a major organ, I would only have a prognosis of about 6 months. So how could annual checkups be helpful in catching it and treating it early? I was not sufficiently informed, which was my fault. Year after year went by. At scans, all checkups were normal. Had I truly won this battle with my errant, dangerous, wild mutant cells? For 22 years, cancer, slept, probably dreaming, pondering how it could conquer me, or at least give me a hell of a fight. I thought our relationship was in permanent truce, but cancer finally awakened to go on the attack. I suddenly had 2 lesions in the liver. Okay. Not knowing what, then the 2 lesions in the liver appeared. Not knowing what my options were, I was really poorly informed about the whole thing, my mistake. My oncologist at the time asked me what did I want to do. What did I want to do? I didn't even know what my options were. So, being a good patient, I said, well, what do you recommend? And he said, I hate to expose you to an invasive procedure like a liver biopsy. I didn't know anything. I didn't know what a liver biopsy involved. I knew nothing. So I said, and so he suggested that I just have I have CAT scans every 3 months, and we'll see if it grows. If it isn't growing, it isn't cancer. Okay. So I said, okay, we'll do that. So every 3 months, I had a liver biopsy, and every 3 months, the radiologist report came back stable, unchanged. Then I went to another doctor for another reason, and he said, you mean to tell me that you have a history of ocular melanoma? You found 2 things in your liver, and you didn't have a liver biopsy? I thought, oh, maybe I should have a liver biopsy. I did have the liver biopsy, and it was cancer. But I want to tell you that the stable unchanged business, another thing I should have done was got the actual pictures, or the reports, actually the reports, which had measurements on them. So I had a CAT scan every 3 months, 4 CAT scans. Each one measured against the previous one, not the beginning. So when I got the reports, I looked at the beginning measurement and the end measurement, and there was growth. This is something that radiologists do even today. They only compare, the ones I run into, only compare with the next previous scan of a similar kind that you've had, and they give you the difference. They don't hardly ever go back, a year before to see what happened. So I knew I had cancer, and in the liver, metastasized, and I also knew that it would have been more than worth it to have a liver biopsy right at the get-go, at the beginning. Okay, so there I am, 22 years into ocular melanoma, and how I dealt with it during that time was denial. I'm very good at denial. My evil twin gave me an indication that he was not there for 22 years. Now 22 years, if you include the year of scanning every 3 months, it's 23 years. So it was easy for me to believe that I was fine. Year after year, 2 lesions were, the year after, when I found I had 2 lesions in the liver, I really didn't challenge my innocent denial with the facts. But trust me, you cannot go back and do things over again, and it just wastes energy to beat yourself up about it. So after all this time, my evil twin was breaking our truce and becoming truly abusive. With the help of my husband, I began to change from being a good girl, compliant, pleasant, congenial, non-combative, non-confrontational, to a very fierce fighter. I don't look too fierce, but I am. But I could not do it alone. I really needed and still need a lot of support, like anybody does in an abusive relationship. As you know, the liver biopsy rendered me stage 4 with a prognosis of about 6 months. So that was counting from then, so when I was diagnosed to now, it's 10 years, more than 10 years. And I also got the application of terminal 10 years ago. During these 10 years, my relationship with my monster twin, cancer, has changed dramatically. No more truce, we are adversaries now. But the relationship has fluctuated nevertheless. Like the proverbial cancer roller coaster, up and down, better and worse, but never good. The cancer has definitely grown, albeit slowly. My attempts to slow it down have occasionally succeeded for a while, but then it goes on the march again. I've had many treatments. Sometimes the treatments have helped in that there may be a regression of some lesions. Sometimes they have not helped at all. There has been a lot of progression. Overall, there's been a great deal of cancer progression, predominantly. But when my doppelganger goes on the march, then so do I. Though I am not medically trained, I've taught myself by reading material, journal articles, referring to assorted information sites when I don't understand something, searching out clinical trials, discussing, mostly online, with others who have ocular melanoma, and connecting with the several wonderful doctors who know about this disease and are generous in sharing their expertise. It has become clear to me that no one doctor, no one institution is conversant with the many approaches being tried for this cancer. One must gather information from many doctors, mostly researchers, read many articles, try to figure out what approach is more promising than another, factor in side effects that one is willing to tolerate, search for clinical trials for which one is eligible, and then make the decision by yourself. However, let me assure you that I am still very much alive, although I have many lesions in many places: liver, lungs, peritoneum, breast, abdominal wall, chest wall, subcutaneous, and more. I cannot remember them all. I am living and enjoying life. Much of my time is devoted to the internet, researching ocular melanoma and so forth. But because my cancer has not yet given me symptoms, I've been able to go on enjoying life. During my cancer career, I've seen my children marry, seen 7 grandchildren born, one of whom is to have a bar mitzvah this fall, which I absolutely, definitely plan to attend. Spent precious time with my husband, published my dissertation 45 years after it was written and accepted. I'm slow. Maybe that's why my cancer is slow. It's a part of me. Traveled, read a large number of books, seen an even larger number of movies and plays. In general, I've taken a great deal of pleasure in life with few restraints, probably with more pleasure than normal people do, and with more exuberance, because cancer gives one an enhanced appreciation of life. Cancer has actually enriched my life in many ways that I would not have experienced otherwise. I've made unbelievable relationships with others who have been affected by this disease. Though I have seen so many of us die during these 10 years, there have been incredible positives. I've made friends not only in the U.S., but also in many different countries: the UK, Israel, Norway, Australia, Canada, and many more. One of these is, or was, a close internet relationship with a cancer researcher from Australia who passed away from OM. His widow, never before having met us in person, has come to see us 3 times, this last time bringing their children. One of my closest friends right now is a woman who has OM metastasized. Her name is Christine Schrader. She said it was okay for me to mention her. Is 3 years behind me, being stage 4 for 7 years now, whom I see many times a year, speak with weekly, with whom you would think superficially I have nothing in common. All the demographics are wrong, every one of them, except for sharing ocular melanoma. But that makes no difference to either of us. We are bonded, and along with the depression, panic, anxiety, and meltdowns one experiences on this journey, and I've been there at all of them, there are moments of humor. Many years ago, on one of the forums in which we OMers communicate, the topic of alcohol and permission to drink came up. Then it occurred to me I should start a new clinical trial. So I started the pina colada trial. This is true. This is true. So every time, Christine was the first one to join. So she and I, we meet maybe 4 times a year, and we have pina coladas every time. So please notice that both she and I have survived much longer than expected. So I think we're on to something. So that's my advice. Another way cancer has enriched my life is my relationship with doctors, many of whom are at the top of their field, yet who are willing, even happy, to help and advise an ignorant patient like me. To be privileged to know these extraordinary people has been one of the highlights of my life. In fact, cancer has brought out kindness in so many people with whom I have been in contact. It's a discovery I would not have otherwise made. It's a blessing. Probably research on cancer has facilitated some of my brain cells staying active so that I can stand before you and be semi-coherent. Cancer has brought my husband and me closer together. We are a fighting team. I could not have come this far without his love and support. For now, the brawl between me and my evil twin is getting down and dirty, but I'm holding my own. Thank you. A list of all my treatments. Okay. Somebody's giving out a list of all my treatments. I thought I wouldn't bother you in this speech. It's boring, it's long, and trust me, it has no magic bullet in it. It's not instructions for anybody to do anything like that. But while they're doing that, I do have one thing I need to do. I have one more message. I hope you can read it. What does it say?
Cancer sucks. Okay, so I have to follow all of that? Really? Thanks, guys. Thank you. My name is Jeff Mark. I'm the caregiver representative here today, up on the panel. Thank you. First of all, I want to welcome everybody to Texas. I'm a Texan, not native, but I got here as fast as I could. We live in Dallas, which is about a 4-hour drive north of here. And we have family in Houston. My sister-in-law is here today as well. And so I'm very happy that CureOM and the Melanoma Research Foundation has put this together with MD Anderson because it's local, relatively local for us. But I wanted to start off by saying that the fact that you all are here, patients, caregivers, etc., especially the ones who are newly diagnosed, you are one step ahead of the game. As you are aware, this is a very rare disease. Many of your doctors, including some oncologists, don't know much about this and may not be able to provide the best options for you. So knowledge is power. You are here, and you're on the listservs and the Facebook pages to gain knowledge as well as to feel a part of a community, which we all are. So I'm tasked with speaking to you as a caregiver of someone with ocular melanoma, metastatic ocular melanoma at that. So I took some notes where I do my best thinking. So, uh-oh, somebody wiped it off. I don't want to know where that went. But luckily I've got my backup notes, so I think we're okay. So let's start with my wife's story. My wife, Deborah Mark, is here, obviously. She is almost 39 years old. I'm about a year older than her, so I'm about to have a big birthday. We have 2 young kids, age 7 and almost 3. Deborah is an elementary school teacher, special ed school teacher, in fact. She is now recently on medical leave, which is great, because she can take the time to really fight this disease. She was diagnosed with ocular melanoma in, good thing I brought this. It's always the caregivers who cry and get sappy, I swear. Last year, in both of the sessions, in all of the conferences I went to, Washington, D.C. for OMF and Philadelphia for this one, all the patients were just, laughing it up in their seminars, and all the caregivers were bawling. We're just, saps. So, bear with me here. So she was diagnosed in July of 2011 with a rather, medium to large size tumor in her eye, ocular melanoma, choroidal melanoma. She had the plaque radiation treatment done in Dallas by Dr. Dwayne Fuller at Texas Retina Associates. Dr. Fuller trained under the Shields in Philadelphia and was part of the COM study, and you saw a little bit of that mentioned in here. So we felt comfortable doing the procedure with him. Now looking back, we wish we had done a biopsy of the primary tumor in the eye. He did not offer that at the time, so we would have had to come to Philadelphia and been treated by the Shields. But nevertheless, we had the treatment done in August of 2011. And then in March 2012, we attended the OMF Patient and Caregiver Symposium in Washington, D.C., where we met many of you for the first time. We learned so much about liver-directed treatment and systemic treatments that we did not know. We also learned about the very few doctors and centers around the country that have any kind of experience in metastatic uveal melanoma. And this was critical, as we wanted to avoid losing critical time if and when METs occurred. Well, unfortunately, it did not take long. About a month, we'd been doing CT scans about every 3 months, and we wanted to get a baseline MRI and a baseline PET CT, full body. So the next month, we got a full body PET CT, that's April of 2012, and it detected the METs in the liver. At that time, liver only. So of course we did an MRI which confirmed it. We immediately sent her information to 2 places, to Dr. Takami Sato in Philadelphia and to Dr. Patel here at MD Anderson, and Scott Woodman as well. We met with Dr. Patel, who was wonderful, and we decided to start with radioembolization, or SIR spheres. Unfortunately, this was denied by insurance as it was experimental and there were other FDA-approved liver-directed treatments available. So we decided we wanted immunoembolization, which at the time was only being performed at Thomas Jefferson University Hospital in Philadelphia through Dr. Sato and his team of interventional radiologists, David Eshelman and Karen Gonzalez, all awesome people, by the way. The interdisciplinary team of Dr. Sato and the interventional radiologist and the MRI specialist reviewed her scans during their weekly Monday morning meeting, and then we began the immunoembolization treatment after that. 1 lobe each month. She went for 4 treatments, 2 on each side of the liver. We found that unfortunately it was not keeping pace with the tumor growth, even though there was some cell death. We asked again about SIR spheres. We were told that it could take up to 2 months to get approval going through the appeals process. The problem is that she could not go 2 months without treatment. So we opted for DebDox, which is drug-eluting beads with doxorubicin, So if you ever see that acronym, DebDox, that's what that is. We followed that up by traditional chemoembolization with the drug BCNU. We actually just came back from Philadelphia this week. We actually flew home a day early. Guess it's when we're sick or home. There was additional growth. They felt that it wasn't working anymore. We've been going to Philadelphia for 3 to 4 weeks, or every 3 to 4 weeks since May of last year. But we weren't going to leave empty-handed. Of course, Deborah's quote was, "I'm not leaving until you put something up my groin." Well, they didn't do that, but they did leave. They did give us a plan. They recommended PHP, percutaneous hepatic perfusion, with Dr. Charles Nutting in Denver. So tentatively, we're scheduled to fly out to Denver on March 18th. Tentatively. There's still some things that have to be worked out. She also has mets on her femur, spine, and some small ones in her lungs. Last August, she had radiation on the lumbar spine, and now she probably needs radiation on her cervical spine. She was also getting a monthly infusion of a drug called Zometa to help strengthen the bones to prevent the bone mets from doing any further damage. With the new tumor on her C-spine, she is switching to a monthly injection of Xgeva. She's also on ipilimumab or Yervoy, and she goes for her 3rd round out of 4 on Tuesday of this week or next week. People have asked me where I got my medical degree from. I have an MBA. I don't have an MD. But you learn very quickly when you're dealing with a serious disease like this. As a caregiver, I work full-time. I help take care of the kids. I pay the bills. I schedule the scans and treatments. I will tell you that Renee from Dr. Sato's team, she's infamous. She is wonderful. But we still do have to manage our scans locally and some other local issues. I try to keep the peace with the kids and try to maintain a sense of normalcy in front of the kids. And that's really the hardest part, because there are so many times I want to do this. I'm a caregiver. I'm not Superman. I've made my share of mistakes, but I can't fly around the world in reverse time to before any of you got this dreadful disease. I wish I could. After a while of friends asking, "How is Deborah?" I remember one of my friends stopped me and said, "How are you? Really, how are you?" I was numb, frankly. I cry, but I try to stay strong, and that made me numb. A caregiver takes care of his or her loved ones with the disease, but a caregiver needs to make sure that he or she is taking care of himself or herself, and that's a message to you all. Why is that important? If you as the caregiver are not taking care of yourself, then how in the world will you be able to take care of others? A sick spouse, kids, etc. Although a caregiver must take care of himself or herself, it is important to filter your reactions to the patient as best you can. The patient is often heavily medicated with painkillers, etc. Memory loss is not uncommon, so conversations and decisions may be forgotten or not retained. Arguments are certainly forthcoming there. "I told you," "No, you didn't," "Yes, I did." Don't go there. Therefore, patience and filtering of your first responses is often necessary. The patient may be irritable, cranky, drugged, in pain, and worried that every pain reflects the spread of cancer to other parts of the body. And by the way, we as the caregivers worry about the exact same thing. We just may not always show it, because we don't want you to worry any more than you already are. If you are a type A personality like me, and pride yourself on analytical and logical problem solving, Patience and filtering your first reactions may be extremely difficult, as it is for me. Oftentimes, a patient's complaining or frustration does not mean that they are asking you to solve all their problems. They simply want someone to listen and express their feelings to. Now, that is also typically true of healthy people in marriages. Guys, men, we want to fix everything when the wife complains. Well, they don't want that. They just want to know we are listening to them and their concerns. I bet you didn't know you were getting marriage advice here today, too. Almost 13 years of marriage, by the way, with Deborah. Thank you. Also, it is important for the caregiver to find someone other than the patient to express their frustrations to, such as financial, the family's future, doing all of the housework and taking care of the kids. The patient is already punishing themselves for not being able to do these things. However, they are the ones with cancer. Normal marriages and relationships have problems and issues that existed before the current crisis and can be greatly magnified. It is challenging not to sink back into pet peeves, but you have to pick your battles and prioritize what's important. You don't have to be a rock all the time. Share your fears and your feelings. However, focus on what is important and think before reacting negatively to anything. And the patient knows you're working for him or her, even if they don't express it. Find someone else to talk to about frustrations, but try to keep it as positive as possible for the patient. Treasure your time together. Also, it helps if you can have other people to help you as well. Deborah's parents, who are here as well, they watch our kids at our house when we travel for treatment. Neighbors and friends cook meals every now and then. Learn to accept others' help. Some people don't know what you need. If they ask, don't be afraid to tell them. Some of our friends asked what they could do, and I told them, cook us a meal every now and then. Our neighbor brings us dinner once a week. We have friends doing the same, and that's a huge help, especially when you have 2 young kids in the house. It's also important to have a sense of humor, both patient and caregiver. We noticed a greater sense of humor, for example, eye jokes, among patients prior to metastasis. For example, when Deborah missed the first 2 weeks of school, she's a teacher again, for her eye surgery, her plaque surgery, she came back after not meeting many of these people, or meeting them for the first time now, and gave everybody these little eyeball hand sanitizers, these hand sanitizers that were in the shape of an eyeball that were sold for Halloween. And it was great. And by the way, that was her first day on the job at that school. And it was a great icebreaker. A lot of, she comes in, her eyes all, red, it looks like meat, and because it was just after the procedure, and it just put everybody else at ease, and it showed her sense of humor. It was a big hit. When Deborah's Mets were detected, she asked her oncologist to see the full body PET scan, and this was a very serious meeting for us, and I was nervous about seeing it. I'm sure she was. The image comes on the screen, and he already told us that that it's Mets and it's in the liver. But this is a full-body PET scan, and the image comes up, and the first thing that Deborah says is, "That is not a flattering picture." My God, I look fat. Okay, and she approved me saying this, by the way. Anyway, I sincerely hope you benefited from our story and this little public service announcement, and I thank you all for the time. Thank you.
We'll go ahead and open the floor if you guys have any questions. There's microphones out on the floor for any of the panelists up here.
Jeff.
Yes.
Do I?
Yeah, I hear you.
Was he 100? My shotgun made that for me. You probably, as I told you earlier today, know more about every phase of it as any one doctor in the country, because they know their little part, like these crazy things today. If you were going to start over, what would you recommend to this group, step by step?
Yeah.
Now, you've been through the 4 big places, And where would you start and what would you do?
Yeah, it's a good question. Number one, we would have gotten a biopsy of the primary tumor in the eye. We would have definitely done that. We would have done a full body PET CT prior to the plaque therapy. So we did a chest X-ray and CT scan, and I believe an MRI, Deborah? Right. So pre-op. No, so CT and chest X-ray and blood work. We should have pushed for a PET CT at that time as well to get a true baseline prior to even the eye surgery. And then we started with CT scans every 3 months. We would have, had we done it again, we would have done MRI probably. And again, I'm not a doctor, but the experts that we've talked to, Dr. Sato, for example, was one of them who suggested MRIs every 3 months and CTs every 6. So we still would have gotten the CT in there, but the MRIs would have given maybe a little bit better look sooner. And then, as far as once the metastasis happened, I think we did. I don't regret the process we took. We sent our scans to Philly, we sent them here, and we tried to do what was best at the time and what was available at the time, and sticking with liver-directed therapy because we knew that at the time that's the only place we had the disease, and it was very fast spreading, as you heard. It was about 9 months from primary to mets. That's extremely fast. So we were really worried, and we really needed to stop that from getting any bigger in the liver. Did that answer your question? Thank you. You're welcome.
Any other questions? Okay. Well, we can, if you guys want to stretch real quick, before we get up, if you want to give them another thanks just for their time and sharing with you guys. I know that took a lot of bravery. The social workers should be in here in just a minute, and then we can break off into the small groups for discussion. Okay. Okay. Yeah, go ahead. I think it's a 15-minute break if y'all want to come back in here.
Yeah. So I think we'll go ahead and take a break for about 15 minutes, and then come back in here, and all the social workers will be in here, and then we'll break off into our individual groups. We're really lucky to have a dynamic team of social workers, all from MD Anderson Cancer Center, so all trained in oncology social work. We're really lucky. If you have not signed up for a group yet, it's not too late, so the sign-ups are out there. And whether you're participating in a group or not, I'll just remind you afterwards, we have the meditation and mindfulness session, which is at the end of the hall from 5 to 5:30, and then dinner is in this room at 6. And just a time for getting together and connecting. We'll have a short celebration of life from 6 to 6:15. Okay? We'll see you back here in 15 minutes.